Thank you very much for that introduction, and thank you for the opportunity to speak to this very distinguished group. So I'd like to point out the real experts here are you, because those of you who have these illnesses or whose family member have these illnesses, live with them every day. You are the true experts on these diseases. But I can bring a physician's perspective, as someone who, as you pointed out, has been doing this for some time, which I hope might be helpful to you. I always like to make disclosures. All right, so those are my disclosures. I do lots of different things. The other disclosure is I came from Minnesota. For those of you who live in this lovely part of the world, I'll point out a couple of things. On the upper right, you notice that not only does my Kia Optima get good mileage, but it was a little cold that morning at 23 below zero. Upper left, you see what happens after a snowstorm, and you open the door of your garage. You're not going anywhere. That light on the lower right is 6 feet high, and that's a view from my office window the morning it was 23 below zero. If you're not familiar, those are called sundogs or parhelion. When it's that cold, ice crystals form in the air and you get refraction of light in this. It's very beautiful as long as you don't walk outside. Now, I can claim to be an expert using the definition of Niels Bohr, who was a Nobel laureate. And you'll see here he said, as quoted by Edward Teller, that an expert is a man who has made all the mistakes which can be made in a very narrow field. So I qualify on those grounds, and I hope I can share some of that experience with you so you perhaps won't make the same mistakes. So I'm going to talk a little bit about the concept of ultra-rare diseases and their management very briefly, the challenges that are associated with studying them, and then say, what is a clinical trial? We've talked about it, but what is it? How do you plan one? Who gets to take part in a clinical trial? When do we learn about it? And how does this data help the rare disease community? Now, we have a legal definition of rare disease in the United States. The Orphan Drug Act in 1983 defined a disease as rare if it affected 200,000 or fewer people. You're all smiling, thinking, are you kidding me? Is that rare? Well, in child neurology, everything we deal with is rare, using that definition. The diseases we're talking about that NTSAD is involved with and that I've studied for many years are ultra-rare, and I propose that we call ultra-rare diseases those that have a frequency of 1% or less of rare diseases. That would mean there would be 2,000 people in the US. 2,000 would probably cover all the diseases embraced by the NTSAD. But this is very important because we need to have a definition of these things for regulators and when we design and execute trials, as we'll see in a minute. Let's say a little bit about managing ultra-rare diseases. And the first comment, which is something which is intuitive and you all know, is that not one size fits all. For any disease or label, it may affect someone in utero. That's the earliest onset of some of these diseases. Before babies are born, they may be affected by this disease. On the other hand, I've diagnosed people in my age group in their 60s who've presented with these diseases. So for any one disease, there's a broad span, and sometimes even within a single family, there's variability. This is very important. So it's not very likely that a single treatment or a single approach to treatment is going to work for everybody, even with one named disease. The manifestations are so variable, we have to take that into account. I also want to make the point that when we talk about treatment, we tend to be focused very much on the right-hand column on this slide, which is so-called disease-modifying therapy, something that's going to change the basic process. That's terribly important. It's a big focus. But I don't want to neglect what's on the left side of the slide, and that's symptomatic management, because this makes a huge difference. You all do it, and sometimes parents come to me, or families come to me and say, well, we're not doing anything. And I'll say, well, now, how many hours a week are you spending on therapy? How much time are you spending working on diet and exercise and other treatments? It's not nothing, and it's important. Nutrition is tremendously important for all of us. Exercise is important. Even if you have a physical disability, there are still ways you can maintain your mobility and your fitness to the greatest possible extent. Therapies are important. Physical, occupational, speech therapy, other types of therapy are very important, and as an advocacy group, it's very I think you've all encountered this. I don't need to tell families the difficulty you sometimes have in getting insurance companies to pay for therapy, because insurance companies think everyone's had a stroke. And if they're not better in a year or two, why are we doing therapy? We have totally different goals with these diseases. So advocacy there and recognizing the importance of therapies in maintaining function is tremendously important. We can manage many symptoms, whether they be seizures, abnormal muscle tone, sleep disorders, with drugs with varying degrees of effectiveness. And one of the things that I've found with diseases I've worked with in the past that's terribly important is to develop disease management guidelines. You want to publish in the peer-reviewed literature the state of the art. Get together the experts so you can say, when you're dealing with insurers and others, here are the published guidelines from the peer-reviewed literature. This is what we need to do. That's a very important tool. It has some scientific value, but as a tool for families and support groups, it's tremendously important. And I want to emphasize again that symptomatic management can actually not only enhance the quality of life, but probably prolongs it. Since I got involved in this field almost 30 years ago now, people do live longer and better because we deal better with nutrition, because we deal better with therapies, because of the development of guidelines. And the best example I can think of is cystic fibrosis, one of the more common rare diseases. I started in medical school in 1976, that's a long time ago, but at that time we were taught that a child born with cystic fibrosis would not expect to live beyond the teenage years. Now, the latest report I saw is that if you're born today with cystic fibrosis, you can expect to live to 50. That's a pretty impressive result, and that's because of the development of expert centers, treatment guidelines, symptomatic treatment. That's before any disease-modifying therapy became available. So this is tremendously important. But of course we are focused, we want to alter the course of disease, and since all the diseases we're talking about are genetic, the ideal answer would be to say, well, if we could somehow go in and replace or repair that single base pair that is abnormal, those single nucleotide bases, we could edit them, that would be ideal. Now, in fact, there's technology now, so this can be done in cells and animals, so maybe that will come for human disease. That's very exciting, and I expect you'll hear something about it in the course of the meeting. Of course, the other thing you can do is take a normal copy of the gene, an unmutated or wild-type copy, and put it into someone's genome. That's what we usually think of as gene therapy. Another approach would be to say, well, if we can't replace the gene, which is hard to do in the nervous system, maybe we can find the gene product, the protein, and replace that. And that's been done for many lysosomal diseases in the form of enzyme replacement. Very hard to do for brain disease as opposed to disease that affects the liver, the spleen, and the bones, and so on. I'm just checking my timer here so I'm not raving on for too long. The other type of therapy we think about are so-called downstream therapies. These diseases have many effects. They may produce inflammation. There's accumulation of molecules which may be toxic. And so there are a variety of small molecules which can often be administered orally and can reach the brain and may also influence the course of disease, and you'll hear about these. And as we've already heard, in our current state of understanding, it's likely that combination therapies, different approaches to therapy which work by different mechanisms working together, will produce the best effect in ameliorating the course of disease. Well, what are the challenges? You already know these things when you've been living in this environment for many years. One is you have to get funding. It's a very competitive environment. When I was at the NIH, there was a big gorilla on the campus. It was called the National Cancer Institute. Okay? Most of the funding went there, because cancer is common. If you include skin cancers, 1 person in 5 in the United States will get cancer during their lifetime. So it's a big problem. It gets a lot of money. It's hard when you have an ultra-rare disease, but we've got some positive developments. One is the Orphan Drug Act itself, more than 35 years ago, was designed to attract interest in this area. And in fact, there's been other rare disease legislation recently, particularly the America Cures Act, which is important for ultra-rare diseases. And pharmaceutical industry is interested. There are people here as a sign of that. So that's a very positive development. We've already mentioned the fact that these disorders are very variable, both in terms of the presentation at different age, and even at any individual age, there's a lot of variation within and between individuals. You've heard about the importance of natural history. What is that? Well, basically that is what happens to people with diseases when there isn't a disease-modifying therapy. And you'd think most of these diseases we're talking about were described more than 100 years ago. Don't we know that? Well, no, we don't know as much as we should. And so the concept, I think, is important that every piece of data, every piece of information about every person with an ultra-rare disease is precious, and we need to capture that, because that is ultimately going to help the whole community as well as help individuals to understand the disease. If we understand the disease and how it typically follows its course, then we will be able to understand when we try experimental treatments whether they're making a difference. You can't tell if you've made a difference if you don't have some idea what was going to happen. That's particularly the case with later onset forms of disease. With the early onset forms, we generally have a better idea because they tend to progress more rapidly. There are ways that, as an advocacy community, you can play a very important role in terms of defining what's important to you as a patient or a family. That's one area I think we have neglected in the past in studies. Now, the Food and Drug Administration and other regulatory authorities understand it's important to hear your voice, so you have to make it clear what matters to you in terms of outcomes in these diseases. You can participate in gathering information. I know you have an initiative here. I've been very much involved with Niemann-Pick disease type C for the last 30 years, and we've actually been lucky enough to, through an international alliance of support groups, to develop an international registry which is funded, controlled, owned by families. It's your data, you control it, and I think that's a very important concept. You've heard about biomarkers. A biomarker is anything you can measure which is abnormal in a disease. It might be an MRI scan, it might be a blood test, it might be something in the spinal fluid. A step beyond that is something we call a surrogate marker of disease progression. Because for many of the disorders which we think about here, the lysosomal diseases, the progression may be very slow. And that's a problem if you want to design a clinical trial to determine, an experiment to determine if an intervention works. Can you do that over 5 or 10 years? That's going to be hard to organize. If it's something you could measure which you know changes in a predictable fashion, that would be very useful to you. And that is called a surrogate marker of disease progression. We're starting to get these for these diseases, but we need more. And of course, because a disorder is ultra rare, there are a limited number of patients who could potentially participate in a study. Not everybody wants to. It's not appropriate for everyone to take part, as we'll see. So having said that, what is a clinical trial? And I think this definition from the European Medicines Agency is very helpful. Clinical trials are experiments. Very important. They are experiments that use human subjects to see whether a drug is effective and what side effects it may cause. That's what a clinical trial is. Before whatever agent you're using in the trial has been tested in this way, it's an experiment. It's not a treatment until we know that it actually does something. Now, when I talk about planning clinical trials here, I'm going to emphasize the so-called classical approach. And some of these things are very hard to do or not feasible in the ultra-rare disease community, but we need to know about it because this is the path, to some extent, we have to follow. The first step is what we call preclinical studies, and these are basically studies that are done in animals, sometimes in cell culture, where you have a promising agent or a mechanism of action, and you're able to show in an animal, it's usually a mouse, but it may be a dog or a cat or some other animal, that the experimental agent you want to use seems to be beneficial in that model of disease. That's very important. It's very rare to go direct to human studies. You usually want to have data in animals, which gives you some idea about safety. Now, there's a caveat there because people aren't mice. You all knew that too without me telling you, but sometimes scientists forget that. And so sometimes something which looks good in a mouse may not work in a human. There may be different adverse effects. So those are important things to bear in mind. Very basic question, which we'll come back to, is if you have an experimental drug, how much do you give? Whatever worked in the mouse may not be appropriate for the human. The metabolism is different. And then you have to reach agreement, and ideally this should be done with the patient community and the researchers, and the regulators will put in their 2 cents worth as well, as what is important what is important to measure, how can you measure it in a reliable fashion? And that's what we call designing outcome measures. And sometimes the regulators will say, well, you better prove that this really works, that it's reproducible. You may have to do what are called validation studies. For common diseases, there are many so-called scales. In neurology, if someone has ataxia, there are many measures of that that have been validated in different diseases. That's sometimes an issue for clinical studies in ultra-rare diseases. And then you have to decide when, how often you're going to give the experimental agent, how much you're going to give it. Can you give it orally? Does it have to be given by vein or into the spinal fluid, for example? Who's going to get this? Who is an appropriate subject for this experiment? How long is it going to last? How are you going to measure the positive effects? These are the outcome measures. How are you going to control for random variation? Because we're not all the same. So there's going to be random variation in people's performance from day to day, and this means you may use a placebo, you may blind the observers, you randomize people either to receive the placebo or the active agent, and then how do you monitor the adverse effects? And all of these things together are called the protocol. And this may be the most boring thing you ever read, a protocol. It's about this thick, everything has to be done, but it's important because before you begin a study, you must plan these things very carefully. You have to think through all the potential possibilities. And if you're the investigator and you're working with the family group, you're not the only people involved. You need statisticians to help you because in most cases you need to decide before the study begins, how are you going to analyze the data? You can't decide this afterwards. It doesn't work that way. You can't say, well, this looks good. Let's do these sums. That's called post hoc analysis, and you can't do that. So you need to decide beforehand what change in your outcome measure is actually meaningful. If you can walk 50 feet in 6 minutes and afterwards you can walk 52 feet, so what? Doesn't mean anything. Maybe what you can walk in 6 minutes doesn't matter anyway. But those are the sort of discussions you have to have. What's going to be a meaningful change? And based on the size of the change, how many people do you need to recruit to your study to have a reasonable chance of picking up that change with a certain degree of certainty? And so to do this, you do something called a power calculation, and that you need to know the N, that's the number of people who will be in this experiment, the theta, which is the size of the effect, how big a change you're trying to measure, and then what's called the p-value. Now, we could spend all day arguing about this. We're not going to now, but we can talk subsequently if you'd like to bring that up because it's a fascinating topic and I'd love to discuss it with you. You then have to decide, before someone takes part as a volunteer in this experiment, they need to understand what's going on. So you need to have consent documents for people who are able to consent. That's 18 and over in the United States. If they're between typically 13 and 18, they have to give their assent, which is also required. We'll talk more about that. And all of this will be regulated by the local institutional review board or boards if you have multiple centers, which you often need for an ultra-rare disease, or the Food and Drug Administration, which is always involved in oversight of these studies. I'm not going to go through this in detail, but for classical clinical trials, they have multiple phases. There's the so-called preclinical phase, which is the studies you do in animals and in tissues. There's a phase 0 study, which you often do in healthy volunteers. In fact, it's often done in the lab of the person doing the study. So if you're thinking about working in a lab, remember you may be an experimental subject because you'll measure the level of the drug after periods of time, after it's been taken orally or after an intravenous injection. You'll see how it interacts with other molecules and so on. That's very important data. Usually doesn't require very big numbers. The phase 1 in a traditional study will be the dose ranging. Again, usually done in volunteers to see, well, if we try different doses, what level do we get in the blood? Do we get side effects associated with that? And that's to try to determine the most appropriate dose. And as you can see, that may be 20 to 100 subjects in a traditional study. Then you do the phase 2, and this is the proof of principle study to say, well, does this drug actually do what it's supposed to do? And in a traditional study, you may have 100, 500 people to do this. And then you will go on to do the pivotal study, the phase 3, which is proof, yes, this does work, it is safe, and that usually leads to approval if it's a positive result. And then there'll be subsequent monitoring, which is subsequently referred to as phase 4. So that's classical trial design. As I said, multiple phases. You need increasing numbers of subjects at each phase of the study. Ideally, you will have a randomized, double-blind, controlled trial. That means when you volunteer to participate in this trial, you will be randomly assigned either to receive the experimental agent or something that looks just the same but does nothing, called a placebo. In fact, that's not quite true. Placebos do something. There's something called a placebo effect, which we can talk about as well. The person or people who are observing your response will not know if you're receiving the active agent or the placebo. So they're blinded, you're blinded, so it's double blinded. You don't know whether you're getting it, they don't know whether you're getting it. And the whole idea is to minimize bias, because if you're an investigator, you want your drug to work. And if you're a patient, you want it to work. So we have to try and remove those natural and understandable biases. And we've already talked about the fact you have to have enough people in the study, you have to have a big enough effect to determine whether this is actually doing what you want it to do. And you use standard statistical methods. But to do this, you need a lot of potential subjects, because not everybody will want to be a volunteer, not everybody will qualify, and we'll talk about that in a moment. You need to understand the natural history so you know what to expect and if what you've done has really made a meaningful difference. And of course, if you have biomarkers, and particularly surrogate markers, that will make it easy. Well, that's great if you happen to be studying high blood pressure or high cholesterol levels, because you've got thousands of potential subjects. And in fact, those studies, although they're expensive, are relatively easy to do. You've got an ultra-rare disease. By definition, not many people have this. There's lots of variability. It's very hard to really match a control group and an intervention group. We still have limitations on what we know about the natural history of disease, and we have limitations on biomarkers and surrogate markers. And the consequence is that maybe these traditional trial designs are not the best ones for ultra-rare diseases. Maybe we need to be more creative. And in fact, there have been some moves in legislation and even lately in the FDA to recognize that fact. So how do we address this? Well, we need better natural history, and I would advocate that as a patient community, you need to be very much involved in taking the lead here. This is your data, you should own it. You need to have registries after approval, that's important. We need to find biomarkers, and that's going to be done in association with natural history studies as well as laboratory scientists. And we need to use alternative trial designs. Okay, let's come to a really important question that I think exercises lots of people in the community. Who gets to take part in a clinical trial? As I've already said, a clinical trial is an experiment, so the subjects in a clinical trial are voluntary participants in a controlled experiment. Now, who's going to be eligible to take part in this experiment is defined by the protocol. And it's very important that you meet so-called inclusion and exclusion criteria. The inclusion criteria include, I'm not listing all the possible things. You need to have the disease. You may say, well, that's ridiculous, we know that. But you have to decide, how do you define that? What test results do you need to prove you have this disease? What stage of the disease are you at? Do you have very early disease so there's nothing you can measure? That might be a problem. Or is the disease so severe you may not be able to take part in what's going on? You need to be a willing participant. You can't be coerced to take part. Now, the worst type of coercion used to be done, and this happened in the past, where people who were prisoners would be encouraged to take part in studies. You know, you'll get a little time off your sentence and so on. That's not ethically acceptable. By the same token, offering people financial inducements is not ethically acceptable, and no institutional review board will countenance that. And you have to have something that you can reliably measure. You would be excluded if you have another disease that might interfere. So for example, in Niemann-Pick disease type C, there's an increased frequency of Crohn's disease. We can't let those people take part in the studies because it's going to confound what's going on. We won't get a clean result. If you're taking part in another experimental therapy, that will interfere with the results. So that would exclude you from taking part in this experiment. And if you have other confounding factors, if you're taking drugs that are known to interfere with the experimental agent, and so on, there can be many reasons. The whole purpose of having these criteria is to make sure that this experiment has the best chance of working and to protect the volunteers. That's tremendously important. The recruiting to a study is controlled. If a pharmaceutical company is running a study, it can't go and approach people directly. This has to be done in a way such that every qualified subject has an equal opportunity of taking part. The consent and assent documents need to be written in a way that you can understand them, and that's terribly important. Institutional review boards emphasize that this has to be clear and understandable. They're very tedious. I've spent time sometimes going through a 50-page document with someone. We're all exhausted at the end, but we do understand what we're talking about as a rule, and that's very important. This must define the risks, the benefits and alternatives, and that risk-benefit ratio is terribly important because that's going to depend on you or your child in terms of the nature of the disease and how much risk you're willing to tolerate. Sometimes as families you're willing to tolerate more risk than the FDA might, so this is a very important message that has to go from advocacy groups to the FDA because they're very intolerant of risk. But I know that in families the assessment often varies. And of course you have the right to withdraw from this experiment at any time. When do you find out the result of these studies? Well, the trial has to be completed or terminated. Sometimes there'll be an interim analysis built into a study and the trial will end. I'll give you an example. A couple of years ago there was a big study of migraine in adolescents. More than 300 children were involved. 2 standard treatments were compared to placebo. The study was stopped early because the placebo was better than either of the drugs. Interesting. Those of us who treat migraine know that well. The data analysis has to be completed. This can take a long time with a big study. As I said, sometimes you could stand outside the building and hear screams, which are the data being tortured by the investigators. Don't tell them I said that. The regulators generally need to see the data, and of course, you have to understand if this is a public company, which is the pharmaceutical company that's sponsoring the study, there are business rules and regulations about releasing information, things like insider trading and so on. So there are a number of factors that determine when information is going to become available afterwards. How does it help? Well, first of all, obviously it establishes if this experiment has been successful or not. Is this proposed agent safe? Does it do what it's supposed to do? It gives you the data you need for regulatory approval, but it's long and complex and expensive. It always takes longer than you want it to take, and it can be frustrating for everyone involved. Well, why do it? Well, one of the reasons, yes, question.
You emphasize a point here that is very important for our families, and that is you talk a lot about the placebo.
Yes.
And many of us as families are concerned that there's a trial that
Yep.
we participate,
Right.
maybe we would have the placebo.
Yes.
And we don't have that luxury.
Yeah, no, I understand, and I'm actually going to give you an example where a study was done without placebo control that's very relevant, but thank you for bringing it up. I understand that's an important point. Just to finish here, when you have a drug approved, it's important because it means that you've got a chance of getting insurance to pay for it. It won't always do that, unfortunately, but it means you've got the best chance of everybody being able to access a treatment that works. The treatment will be monitored after approval, which is also important in terms of safety. There have been some drugs where adverse effects haven't shown up till after approval. And if you've got one drug approved for a disease, it provides a roadmap for all of the rest. And I'm just going to finish with 2 examples about how things might not go right and how they might go well. This is a study that was a big deal in the Niemann-Pick C community using intrathecal injection of an agent called cyclodextrin. So these patients had spinal taps every 2 weeks. This was a traditional study design, so patients were randomized, double-blind. They either got the experimental agent through a spinal tap, or the children had a fake spinal tap, a sham spinal tap where they were sedated. They had a prick made in their back, so it looked as if they'd had the spinal tap performed. 51 subjects. They had very complicated compound outcome measures measuring various clinical things. After 52 weeks, and according to the sponsor, $100 million, there was no difference between the active group and the sham group, and yet everyone believes that this agent works. And I think the problem was they used a traditional trial design, and if they'd used an alternative, more appropriate trial design for an ultra-rare disease, I think we would have got a different result. This is still before the FDA, we don't know. But I think the problem was outcome measures. I think it was trial duration. I think it was selection of subjects. So I'm just emphasizing that trial design is tremendously important and that the traditional trial design doesn't always work. Here's a beautiful example where an alternative design works, and this addresses your question, sir. NCL2, type 2 neuronal ceroid lipofuscinosis, is a very rapidly progressive, typically infantile disease, which is very similar, for example, to infantile Tay-Sachs, some forms of Canavan, GM1, and so on. So this study was non-randomized, single-arm dose escalation clinical study of 22 symptomatic children with CLN2, and the FDA allowed the investigators to compare them to 42 unrelated patients from a natural history cohort that was independent. So they said, look, we know from these 42 patients, all of these children are going to progress rapidly over this period. We expect that, we know that. The children had to be at least 3 years old. They had to have some symptoms, not no symptoms. And then when they performed the study, the treated patients had fewer declines in walking activity compared to the untreated patients. The drug was approved. So the whole point of this is, yes, there is a traditional approach to clinical studies, and I wanted to present that to you. It works for common diseases, but for ultra-rare diseases, it may not work, and the FDA has shown itself open to alternative trial design. So that's a very important thing to know. Those are very important considerations, particularly for the youngest children. I think we know that. Maybe a little different for older individuals. So to summarize, ultra-rare diseases, as you all know, pose very special challenges in drug therapy development. Patients and families need to be in the forefront of the natural history process as well as the design of clinical trials. You know best, if you're a patient yourself or if you have a family member with one of these diseases, what's really important to you in quality of life. So I think it's tremendously important that you have a voice and you make your voice heard to the regulators. They will listen. I was just at a patient-focused drug development meeting for the FDA for Niemann-Pick disease a couple of weeks ago. The regulators are prepared to listen to you. They understand that you know more about this disease than any of the rest of us, or these diseases, I should say. The traditional trial models are often not suitable or feasible for ultra-rare diseases. So this is something that is finally being accepted, but again, you need to be strong advocates for this. The Congress has directed the FDA to use alternate trial models and approaches, and they've passed legislation, particularly the America Cures Act, which is important. Be an advocate, let your people in Congress know your feelings about this, and recognize the alternate trial designs have been successful and they've led to approval. So again, I want to thank all the patients and families who've taught me a great deal and who continue to be my teachers, as you are, support I've had from some philanthropic funds at Mayo as well as other organizations. And I will stop here. I hope I didn't take too long, but thank you very much for the opportunity to speak.
So Mark was terrific as always, so very little to add in terms of the landscape here, but I thought I'd add just a few comments and then gradually also transition to what I think this session is about, which is the family and patient perspective on what you said. But maybe just as a footnote that here we're seeing, you know, in a time of terrific change, now modalities in gene therapy come about that are by their very nature challenging traditional trial design. These are treatments that are one and done, which has advantages and has disadvantages, right, that you can't modulate as you could with small molecule or enzyme replacement approach. And so I thought I'd ask you first about that, and maybe to add on another second question that you touched upon in terms of patient selection. And as we are designing trials around gene therapy and other modalities, you know, I think it's amazing to see, you know, the potential and hope for these treatments to transform patients, but it will not help all patients the same way. and many might stand to have more risk than benefit. And so maybe to touch upon those two things, Mark?
No, I think you raise a really important question. I'll give you an example. I have a couple of brothers I diagnosed again with a form of neuronal ceroid lipofuscinosis type 6, who both had gene therapy. Now, both boys had moderately severe disease, and the positive side is this: they're both still alive. They're still going, but they have very significant neurological problems. And so I think that brings up one issue, and that is, if gene therapy does what you want it to do, and that is to basically begin to produce a normal gene product, it means from that point onwards, in theory, you should stabilize the disease. But what you can't do with gene therapy is fix what's already broken. So I think that comes back to your point that timing is critical. And of course, the ideal time to treat anyone with any of these interventions is before they have symptoms. Now, that's a problem if you're doing a clinical trial, because then what do you measure unless you have a surrogate marker, or unless you, for example, have a mutation where you know there'll be progression. So in another example, Krabbe disease, which many of you I'm sure have heard of, if you have a 30 kilobase deletion in the gene, then it's almost certain you'll have early infantile disease, which will be very rapidly progressive. So in a situation like that, were there to be gene therapy available, you might want to administer that right away. But I think the key point is that gene therapy has tremendous promise, and I think it's starting to be realized. There are a number of problems, and Florian's an expert, and I won't trespass on that field, but we know, for example, you not only want to express the normal protein, you want to express an appropriate amount, because now people can do gene therapy, so you produce a huge amount of protein. And I think at first people thought, well, you can't have too much. Well, it turns out you can, and that can be damaging. So these things are not without risk. There have been problems in the past. For example, if you're taking a normal gene and you're putting it into the genome, you can't just put it anywhere, because you don't want to, for example, put it in the midst of an oncogene and give someone cancer. So there are lots of complex aspects to this that, again, I don't pretend to be expert in, but I think the key point is to remember, as we say before, not one size fits all. Gene therapy has tremendous potential, but I think it's important to remember that you start the clock, as it were, at the point where you are when you receive the gene therapy. So the outcome is going to depend very much on what your functional level is, how much reserve capacity you have at the time you receive this therapy, if it works perfectly. That's very important. I guess there's another concept I didn't talk about, but which I think is very important in these diseases, and that's the idea of reserve capacity and a symptomatic threshold. You know, when we talk about lysosomal diseases, we often talk about the age of onset, which is very important, because in general, the earlier the age of onset, the more severe the course of disease progression. But what's important to remember is if you have any specific symptom in any bodily system, that typically means that the reserve capacity of that system has been largely exhausted. I'll give you an example that we all learn in medical school. If you take mice or dogs and you remove one kidney, 50% of kidney function, what happens to the blood tests? Nothing. Your kidney function tests are normal. You take out 1.5 kidneys, nothing. You've got to take out basically 1 5/6 of the 2 kidneys before you get an abnormal blood test. What that means is most of your reserve capacity has been exhausted. And we think the same thing happens, it's harder to measure, but the same sort of thing is happening in the nervous system. If you have an unsteady gait, it means that there's been a substantial amount of damage to the cerebellum and its connections, and so on. So that's a very important concept to bear in mind, and it influences what we might expect from any particular therapy. I think probably people are aware of that, but I did think it was a point that's worth making.
So I'm going to start to have people prepare questions or think of questions, but just in preparation for that, we're going to try a little thought experiment here, Mark. So you've been in the field a long time, and I want to since you've seen many therapies come and go, if you were to put yourself in the position of a patient with a late onset disease,
Yeah.
you know, we've been coming here every year for years. We're waiting for all this preclinical data to come about. It's all taking so long. What should I do? What should I be waiting for treatments? What if this treatment I will not even fit the eligibility criteria of this trial? What else is out there? How much can I do on my own? What would you do, Mark?
Yeah, okay. Well, he knows how to ask tough questions, doesn't he? All right. Well, number one, I'd be frustrated because I'm human like everybody else. Number two, I guess what I would ask is, what do we know about this proposed treatment? You know, is there encouraging data? I mean, has this been tried on mice or cats or whatever animal model? Has it shown promise? Does it seem to be safe? Does it make sense, as I understand it, and as it's been explained to me? And then, okay, two options. One is I'm eligible for the trial, and then the question is, how do I see the risk-benefit ratio? What's the downside of taking part in this? And if I think the downside is small, and I'm concerned that things are getting worse, then I would probably say, yes, I'm going to sign up for the study if I'm eligible. What if I'm not eligible, but I think, gee, this might suit me? Well, there is an alternative approach. And again, there's some legislation relative to this that applies in different states as well as federally, which is basically to give you an opportunity to try treatment. It's legislation that has no teeth because it can't force anybody to make a drug available, but there is such legislation around. And the approach in that case is sometimes that sponsors of trials will offer what's sometimes called expanded access. And that means if you have a promising therapy, you may give the opportunity to individuals who don't meet the criteria for the formal protocol to receive access through what's called an individual IND, or investigational new drug application. And I think that's something that's basically an uncontrolled experimental study. You're the subject. It's called an individual IND, an IND. Now, sometimes you use the term compassionate use. I have to tell you, you can read, if you have a lifetime, the entire Code of Federal Regulations, the CFR, and you will never see that word. Okay, the term is an individual IND, and it's actually important to know that terminology. Sometimes people use the term expanded access. That can be something that if it's offered to you, if it were offered to me and I were in that situation and I thought, this sounds promising, then I would try. Remember, you are taking part in an experiment of one. You're it. And you don't just take this like candy. You are subject to the same controls of an institutional review board and the Food and Drug Administration. They're going to be monitoring what you do. That's actually a good thing because that's designed for your safety. So if I were in that situation where I thought something was promising, but I wasn't eligible for the trial and expanded access was offered, then I would probably sign up for an individual IND. But again, that's going to be very much an individual decision based on what you understand, and that's where it's very important to have an honest broker, someone who can give you an objective information about whether this makes sense and the quality of the data that you're going to use to make your decision. It's never going to be perfect. There's no such situation. So it means you make, and I think this is a very important point to make, whether you're talking about taking part in a traditional or an alternative clinical trial in a formal protocol or an individual IND, you make the best decision you can with the information that's available. That's what we do in life generally. I mean, who knows all the information, you know, right? If we knew that, our investments would be mine would be a lot better than.
No, I just want to mention that there's also small group INDs, not just individual.
Yeah, you can, yes. The point that was made was that sometimes they're called individual INDs. Sometimes they can be issued and they can be done in groups. That's absolutely right. They're still actually the individual, they're issued for the individual, but it may be possible using that at one site, because there usually has to be an investigator who's responsible, and that investigator may enroll multiple subjects. You're absolutely right. But technically, it is still an individual IND, and your data has to be monitored individually. But yeah.
Who's question?
I was going to ask, because it's an individual IND, where does the funding come from if they're not eligible?
In the case of individual INDs, it's typically going to be the sponsor who makes it available. So when it's an agent that's being studied by a pharmaceutical company, they'll usually fund that. But it's a very good question, because I have sometimes patients come to me and say, this sounds like a good idea, can we try it? And I'll say, well, I'm willing to do the paperwork, to write the protocol, to fill in the 1572 form, to go to the FDA, but we have to find someone to pay for it. And not only pay for it, but actually get access to it. Because there's another issue which is not always recognized. If you're talking about an experimental agent, there may not be that much of it. If a startup company, for example, is making an experimental drug for a study, they may simply not have enough to supply to individuals who are not participating in the study. There's not an unlimited amount available. It's not like getting aspirin, for example. And that's something I think sometimes we forget, but that's actually an important point. So there needs to be drug available, and there needs to be a way of funding it.
Yeah, I mean, and you've probably seen this in CLN7, Mark, recently, an antisense oligonucleotide developed for a single patient.
Right.
I mean, that raises the same questions. Please.
Okay, I have a question. If there are positive results during a clinical trial, how long does it take the FDA to approve it for the general population?
Yeah, that's a wonderful question. I wish I had the answer to that. I've often criticized the FDA, but let me defend them. The FDA sees its most important role as protecting the public, and so they do take some time with their deliberations. The trouble is they're just taking too long. So I can't give you an answer to that. There are certain circumstances where, when a very strong case is made to the FDA about a pressing unmet clinical need, and this is where advocacy is important, they can hasten the process. And so it may be done maybe within a month or two. That's theoretically possible, and occasionally that can happen. It's an excellent question. I can't answer it because I'm not the FDA, and I'm not sure even the FDA would answer it. Based on my experience, it usually, if you have a positive pivotal trial result, it usually takes many months for the approval to come through, but they are improving. And I think, again, with recognition of the urgency of the need, that they're doing a better job. And again, I think the role of advocacy groups is critical here. And sometimes now, sponsors will go to the FDA with the results of a trial and bring along patient advocates to say, this is really an urgent unmet need, and they listen to that. So again, I just want to emphasize how important your voice is in this process. It's a very good question, thank you.
Just a question. You mentioned in your exposition the other therapies, the alternative therapies, as physiotherapy, speech therapy for swallowing, and then occupational therapy, and nutritionists and nurses and all the therapies we use. And you also mentioned that the trial, the medicine, will give the solution for the problem for now on, but will not correct what was broken. And I would like to ask if these therapies will not also help to fix what was broken.
Yeah, I think if I understand, what I was talking about was symptomatic therapies, which means these are things which are not known to alter the course of disease based on their mechanism. But in fact, what I did say at the end, and I think that Florian can speak to he has some data, I think, in some of the NTSAD populations, that in fact, if these life-enhancing approaches are used, they can actually not only improve the quality of life, but duration of life. So it means things like recognizing and treating infections early, making sure that you optimize lung function, making sure that you optimize mobility, dealing with nutrition. For example, we know in people with neurological impairment, the major determinant of survival historically has been the ability to swallow. And now, because when I was starting out, a gastrostomy was a big deal. And sometimes they tended to be left late. I had patients who didn't survive a gastrostomy, which is terrible looking back 35 years. Now it's a routine procedure, and it's something that can make a tremendous difference to people's quality of life and duration of life. So these are things which don't directly affect the disease mechanisms, but can definitely have an influence on the duration as well as the quality of life. But again, Florian, I think, has some data to speak to this.
Yeah, about 10 years ago, we worked with NTSAD to look back over time at the database and also the literature and see how survival had changed within infantile patients with Tay-Sachs-Sandhoff disease. And what we found to our surprise is that really survival had improved over the decades, despite there not being a disease-modifying treatment, and it was largely due to things such as G-tube placement, improvements in medical hygiene, and the like. So I do think there's an important point to that. To maybe just touch upon another part of your question, which is if you institute symptomatic therapy and it enables more function and better quality of life, you are using your body more. And as neurologists, we like to say, if you don't use it, you lose it. Are you actively, using your nervous system in a way that helps propagate health? There is a sweet spot there where you can still be disease modifying. So I think there's some truth there too.
Absolutely, yeah. Yes, sir, the question over here. Oh.
Yes, hello. You mentioned that gene therapy or whatever other medications that are being developed could stop the disease, stop the progression of the disease, but we are not hoping for a real reversal. Do you know about any technologies, and I would include some devices that can help to compensate for lost functions, as well as there have been a lot of talk about stem cells and so on, as that also can, again, do a partial compensation or reversal of the disease. And once again, I want you to talk also about devices, because there have been a lot of information about the different programming that could compensate. Okay?
Yeah,
Thanks.
thank you. Well, you're right. I mean, there are some devices that can certainly compensate for loss of neurological function. The best example of that is the cochlear implant for deafness. It actually has turned out to be quite an effective device. Like any device, it has drawbacks because you increase your risk of meningitis, for example, if you have a cochlear implant. But overall, it's quite effective. There are interfaces being developed now to try to compensate for visual loss. People, of course, have been working for some time on limb replacements with interfaces with the nervous system, for example. So it's a very active area of research. In general, those types of devices are going to be most effective if you have a focal deficit or area of damage in the nervous system or a specific modality that's involved. Compensation is much more difficult when you have diseases that are globally affecting the brain. It doesn't mean there may not be a role, but it's going to be that much more challenging. So I think, yes, we already, as I say, the best example I can think of is the cochlear implant is something that will actually, it's a technology that will replace a lost special sensory function that's quite effective. There are more along those lines. As far as stem cells go, I actually had reason to communicate with a former colleague from Columbia who's in California now leading the stem cell program there. And I guess I like to use this analogy with stem cells. A normal stem cell is like a seed that you plant in fertile soil. The fertile soil is the developing embryo and fetus. And it develops effectively because it's sending signals and receiving signals. It's an interaction with the cells around it. It's a very dynamic process. And in the case of the nervous system, you're talking about the need for these cells to make thousands of connections so that they will develop in the most appropriate way. Very complex process which I don't think we fully understand. The problem with stem cells is that instead of people can certainly engineer human embryonic stem cells now. You can take the stem cell, but instead of planting it in fertile soil, you're putting it on stony ground. And so, for example, there was a study of human embryonic stem cells that were directly implanted in 8 different sites to the brain in children with forms of neuronal ceroid lipofuscinosis. The procedures technically were fine. These were children with very advanced disease who weren't expected to survive, and when they did pass away and their brains were examined, the DNA of the cells that had been injected was detectable, but there were no cells. The cells simply didn't survive in that environment. So one of the approaches that's being used now is to say, well, what if we could take these stem cells and program in all the things they have to do to develop normally? And people are working on that. But that is a tremendous challenge. People are working on it. I don't think it's, from my discussions recently with folks working in the field, it's not really ready yet for prime time in the nervous system. And that's because the nervous system is so much more complicated. When it comes to stem cells to replace the bone marrow, quite honestly, your bone marrow can be anywhere. It doesn't matter. It will work. It doesn't have to be in the bone marrow cavity. Neurons have to be the right sort of cells in the right place, and they have to make all the right connections to function normally. It's like that definition of neurology being like real estate. It's location, location, location. Everything we do, if you work with neurologists, we know what are your symptoms, and we're then thinking, well, what part of the nervous system is involved? How can we figure that out? It's because this location of cells and structures in 3-dimensional space and the way they communicate with one another is absolutely essential to the function of the nervous system. So if you have a brain that's been damaged by disease and you're trying to put cells in, they've got to get to the right place and make the right connections. That is a huge challenge. I'm sure someone is going to figure out how to do it. I think there are going to be several Nobel Prizes awarded when it's figured out, but we're not there yet. But that doesn't mean it won't happen.
Yeah, so maybe two quick things, Sophia. So I think, as alluded to, disease is dynamic. I think the more retained function there is, the more reversibility is possible. Yeah. And the second, what Mark alluded to is, I do think with us understanding more of the crosstalk now between bone marrow and brain, we've at least learned through gene therapy trials that use the hematopoietic stem cell niche that we've over time seen even treatments show some reversal, but particularly in the metachromatic leukodystrophy trial where nerve conduction velocity is improved. So I think it's disease stage and it's also compartment. If there are certain parts of the nervous system that might still show reversal. So I think there's...
Yeah, I think I should modify what I said, and that is, when you think about symptoms, they're really reflecting the number of neurons that have been lost, and we can't replace those with any of these therapies, but it also, the reversible part or rescuable part is the neurons that are sick and might potentially be rescued. So that's right. I guess there is at least some potential for improvement. What we can't tell directly is how much a specific symptom might reflect neurons that are irreversibly lost or damaged and those that might be salvageable. We don't have ways of measuring that yet.
Other questions? I've been reading a great deal about neuroplasticity and neurogenesis. That there is certainly talk I don't know about evidence, but talk about various interventions, designed interventions to increase neuroplasticity and even neurogenesis. Is that something that you feel prepared, or is it relevant?
No, I think it's very relevant. There are stem cells in the brain. That's actually been known since the work of Pascal Rakic in the 1970s. Most of them are in the temporal lobes, which is a very active area in the brain, probably because of its involvement with memory, and it's been very much studied because of its involvement in seizures. There aren't enough stem cells in the brain, though, to replace significant areas of tissue loss. And that's really the basic problem of most neurological diseases. You know, if you have damage to almost any other tissue in the body, there's fairly substantial capacity for repair and replacement. There is some in the brain, but it's much more limited. So that really speaks to the issue of neurogenesis, the ability to generate stem cells. So there are some, but not really enough to deal with significant injury. Plasticity really refers to the ability of different parts of the brain to subsume different functions in the face of an insult. That's been probably best studied, and it makes most sense to be effective where you have focal injury to the brain. So if you've had a stroke where there's one part of the brain that's been damaged, then adjacent areas can rewire and take over some of that function. There's been a lot of work done in that at the NIH and other places showing you get some degree of rewiring. The younger the brain, the better it is at doing that, which again is an observation. But for diffuse diseases, that's a much bigger challenge.
So
If there were a treatment that curtailed the progression of the disease, then might not there be a possibility of neuroplasticity?
Yeah. So the question was, if you had a treatment that could curtail progression of disease, could there be a bigger role for neuroplasticity? Yes, is the answer.
Would someone like to ask one more question? The last question.
Thank you. This has been incredibly informative and educational and just at the perfect level, I think. My last question is asked as a parent. If my child were still alive, how can you just help us, how would you have us think, hold the hope with that complex, long, uncertainty experiment language at the same time?
Well,
just how to sort of think, hold it all, and what might be possible, just so that we have a sense of how to be both realistic and hopeful, because it's so personal.
Yeah, I think that's a very insightful and extraordinarily difficult question. I can certainly reflect on my experience with, for example, the Niemann-Pick community, because when I first got involved, we actually did a study of cholesterol-lowering drugs, which we thought might be helpful, which weren't, although cholesterol was and is important. And so we've sort of evolved through that stage, and I guess there were a couple of points I would make. One is I would be tremendously encouraged by the fact that there are now several potential treatments. I think that's exciting. It's a wonderful development, and that's going to be the way forward. I think that I would, as a member of the community, be active as an advocate, because I think the way that we're going to improve the process is going to be largely through the voices of patients and families. The FDA is listening. The Congress has heard, and believe it or not, I actually testified before them last year, and there was bipartisan agreement. I know, nobody can believe it. I couldn't either, but it can actually happen. People are interested in supporting folks with these sorts of diseases. So I would take that as something I think should be very empowering for this community, to take that forward. The FDA has shown encouraging signs. They have approved an agent for a disease very similar, another lysosomal disease, based on an alternative trial design, using natural history data without a placebo arm, and it's led to approval of a treatment. So I would be very encouraged by all of these things. I'm not minimizing the difficulties, and we know that of the alternatives that are available, they're not all going to work. I don't think they will. I think it'd be wonderful if they did, but realistically, they're probably not all going to work. And even if a therapy works for one type of patient, it may not be equally effective for another. So this really is an early, it feels like a late stage, but it's an early stage. It's a bit like Winston Churchill at a famous speech he gave after El Alamein, where he said, this is not the end. And he said, it's not the beginning of the end, but it's the end of the beginning. I think we're at that stage. I think this is the end of the beginning, but there are very exciting prospects ahead. Those prospects are accompanied by challenges of their own, but I think this is a tremendous opportunity to act as a community for advocacy, to make sure your voice is heard. What's important to people who have these diseases and their families, make sure you have a place at the table. That's going to be important, and it's going to improve, and I think ultimately accelerate the process.
So Blythe, if I understand your question correctly, it's about how do I set my expectations right? And in addition to everything Mark said, I think often we look at treatment as the magic bullet, and in reality, it's a process. And it's a process of putting many pieces together, and there's meaning in all of those steps. And I think if you're contributing along the way through patient experience, through advocacy, through supplying samples, biomarkers, helping inform all of that, you're contributing. And as we have treatments, as Mark said, they probably won't be perfect. They'll need further optimization. And this is where I think all of your experience comes in as so valuable. So I think there's real legacy here for all the patients.
Why isn't it within the medical community, like cancer and MS and any other disease that every doctor knows about? Why doesn't every doctor know about Tay-Sachs, late onset Tay-Sachs? All these people here with late onset, if you hear their stories, they've been taken from doctor to doctor, given multiple diagnoses, none of them the correct one. So why isn't it spread through the medical community that when people are coming, why aren't they testing for it?
Well, it's a great question. The answer is that those of us who are interested ask this question too, but we're interested. And the fact is, as you probably know, the estimates are there are 6,000 or 7,000 rare diseases. No doctor knows them all, and no doctor is ever going to know them all because they're increasing at a rapid rate. I think the answer is we educate people. Doctors have to know, first of all, there's something wrong. They have to listen to families, which is probably the most important message I give to my residents. Listen to mothers. They know more than you do about their child, usually right, and I'm sure there are many people here will have stories about how their concerns were not addressed appropriately. So that's something that's realistic we can do in the medical profession. I think what's ultimately going to make the difference is technology. And the comparison I like to make is with the airlines. Now, I had a fairly horrendous journey here from Minnesota, but I got here alive through the bomb cyclone. And that's because the airlines have engineered their systems so that it's the safest way you can travel. You're taking your life in your hands getting on the interstate compared to flying. There's a multiple in terms of the risk. It's engineering, and I think in medicine we can learn from that. What we need to do is engineer the system so we don't count on individuals to remember arcane things. Some of us like to do it, and we think we're good at it, but really we need to engineer the system so that we use this growing technology. And I believe that whole exome and eventually whole genome sequencing is going to become universal. And I would bet, if I were here in 20 years' time, I'd be really quite old then. If I were here in 20 years' time, we'd be having a very different discussion. I think the diagnostic odyssey will be a thing of the past. I think everyone will know will have had their whole genome sequenced. It's technically possible to do it now. The difficulty is in interpreting it, but that will get better. And so I think that this problem of delayed diagnosis is going to be fixed by technology. Believe me, I think everyone here has spent a lot of time educating other doctors with varying degrees of effectiveness, but we're going to be successful with engineering. That's going to bring problems of its own, because then we're going to have to say, what do we do with all this information about carrier state and so on? But that's another discussion. And I'm going on too long now.
We're going to hold all of the rest of our questions for either the small groups or for our speakers will be around at lunch and after lunch. But I want to thank both Dr. Patterson and Dr. Eichler for joining us this morning. And then before we break up into our groups, I'm going to give a super, super fast, because we're a little bit behind in the research is exciting, plug for the PINs. So hopefully you're familiar and you've heard of these, but these are Patient Insight Networks, or PINs. And they're a type of registry. So we've talked a lot today about how gathering information from families and patients is so important so that the research community and the FDA can know what's meaningful and how to know if therapies will impact disease. And so the PINs are a really great way to do that. They're not a traditional registry where physicians or researchers are collecting data, but they're online portals where patients and families can share their information. So it's really a global patient community. You can sign up online, and I'll show you very quickly a little bit of their information, but they have questionnaires that are already existing, and then researchers can also ask for specific questions. So if you were to sign up for the GM1, for example, you may get a notice that there's a new questionnaire and they're asking information. So right now there are PINs for all of our diseases, and very quickly the numbers in parentheses are the numbers of people who have signed up for both infantile and juvenile GM1, infantile and late onset GM2, and Canavan disease. And we've been doing this working with partnering organizations, so with all of the other organizations who are involved in caring and raising awareness for these diseases. So I would strongly encourage you to take a look at these. And this is very, very quickly a little bit of the information. I don't know how well it shows, but this is for some of our different disease groups. The kinds of questions I ask are about sort of how did you get to a diagnosis? So some of those questions are things like, how old were you when a diagnosis was made? How many different doctors or what types of doctors did you see? So we can gather some of that information. Some of the questions, a couple of those also are information on the impact. So they may ask questions like, how often do you go to the doctor? How many different doctors are you seeing? How often have you had these other events, like being in the hospital, having surgeries, those types of things. So these are direct and easy ways where patients and families can start to share information. And this information can be shared with academic institutions, with patient organizations, and with researchers. Always, it's important to say, in a de-identified way. So when you enter your information and it's shared, this is the type of information we get. There are 36% of people saw the doctor within the last year, but it doesn't say you saw the doctor twice a year and you saw it once a year. So no one knows about you specifically.